All right, teacher, I will start admitting them.
Welcome to the beginning of your future in healthcare. Feel a sense of belonging, no matter which path you take.
I chose the arts because it covers subjects like business, marketing, communication, psychology, and how to apply them within the world of healthcare. Our classes involves a lot of analysis, interpretation, discussions, and sharing perspectives, which I really enjoy.
So class, what do you think?
I love that we're given the freedom to be more hands-on as we collaborate in teams to conduct different experiments for different subjects. These immersive lessons have made biology, maths, and chemistry all the more exciting to me.
Discover how you thrive best as your passions and interests come together.
We aren't just memorizing facts and formulas for our exams. We're being taught to use this knowledge for problem solving and critical thinking, which is crucial for anyone pursuing medicine or health sciences.
Being in an active learning environment helps us create a training ground for us to be more well-rounded in handling different types of scenarios. Our assignments not only enhance my creativity and communication, but it also helps me in my practical skills and analytical thinking as well.
Know that you are on the right path when you feel good about your decision.
Being here has been an essential transition after secondary school to mentally and emotionally prepare me for university. It's become clear to me how this foundation in science offers so much more than just a direct pathway to undergraduate studies.
The more I learn about the social sciences from the Foundation in Arts program, the more I learned about myself as well.
Now we're ready and excited about our future in healthcare.
All right. Hello, everyone. Good morning. Very good morning to all of you students. Thank you so much for joining on a Saturday morning. Yeah. And we understand that next week is all of your science subjects. SPM exam, very, very important, especially if you are looking towards to pursue in health sciences or in any science courses for your your studies after high school. So today's session is very, very important. We are going to be touching on biology. So maybe before we start, there's still not many students in this room yet, about 50 odd plus of you. Make sure that you share this link to all of your friends, your classmates who are taking biology, because today we have Chigu Gan here with us. And Chigu Gan has so much experiences in teaching biology, more than 30 years, and she's also book author for many of the reference books and also textbooks that you guys are using today. So in the past, we have been engaging with Chigu Gan, I think for the past 3 years, right, Chigu? Yeah. So we've gotten very, very good feedback. So make sure you take down notes, make sure you screenshot her suggestions and tips and tricks on time. And we just have a small little note for everyone here today is please keep your questions towards the end of the workshop. Okay, so if you have any questions, make sure you jot them down and then ask them at the end of the workshop via the chat box or unmute yourself. Yeah. So because Chigu Gan does not want to be disrupted during the session to affect her train of thoughts. So this is very important. There's a lot of things that Chigu Gan wants to cover, and basically all of us here wants all of you to succeed in the biology paper next week. Okay, just another reminder. Right now it's 10:04. Today's class will be until 12. unless Chigugan requests to add on a little bit more time, then we will add on maybe just 5-10 minutes. Then later on in the afternoon, we have chemistry paper with Chiguli. Yeah, so I believe you guys are also taking chemistry paper as well. So make sure to join us in the chemistry session later. All right. So I just want to one last reminder, one last encouragement to all of you is please share today's link to all of your friends. This class to all of your friends. It's going to be very, very valuable. Okay. So without further ado, Chigugan, are you ready?
Okay, yes.
Yay. Okay,
Okay.
Chigugan, would you like to
Okay.
turn on your camera? Yeah,
All right.
fantastic.
Hi.
All right.
Okay, thank you, Brian. Hi, everyone. Okay, I'm Cikgu Gan, your host today in bringing you whatever that is important for bio this coming next week. Okay, so I have 2 hour session. So the first hour I'll be doing Form 4 and the second hour I'll be doing Form 5 topics. So the first hour, after I finish that Form 4 session, if you have any question, then only you ask me. Maybe we'll take 3 minutes to ask Q&A for the Form 4 section. And then I will move on to the Form 5 section before we end our session today. Okay, teacher would like to say that actually today session is actually teacher is not teaching. Teacher is pointing out what is possibly important things that you need to focus on. Okay, so I think I no need to introduce myself. Brian has done that. So we shall begin now. All right. So let's start with what would be the important things that you need to focus on this year? So for the first one, teacher feel that maybe this year they will ask about tissues. So the tissues can be human, can be plant. Okay, so I'm going to do just a short, brief introduction on the tissue. So from the tissue here, you can see there are 3 types of tissues. Make sure you know how to name each of the tissue. You know how the diagram of tissue L look like and what is the kind of tissue is tissue L, tissue M, and tissue N, which means that you need to know specifically the drawing of the each tissue. So here, tissue L is skeletal tissue. Okay, skeletal tissue look like small cells as stacked on top of one another. And then L is skeletal. So skeletal is usually like logs like that. They are piled up on top of one another. And M is cardiac. So they have linkage. You can see that's linkage here. So they are different from tissue L. Okay, so must be specific. L is skeletal, M is cardiac, and N is smooth muscle. Okay, smooth muscle. All right, what is the organelle found in abundance? Organelle is also cell component. So here in abundance, so you see the word more there. It means that there are more mitochondria. Can you see the word more is bold? So that means you have to say more energy. Every time you see the word abundance, you must use the word more because every cell has mitochondria. But why is this cell different in the sense that it has more abundance? So you have to say more energy. Now, for muscle, it will always be to contract and relax to produce movement. Okay. Next, how does exercising able to increase the rate of activity in tissue M? Now, look at the second sentence, which is to change the rate. So you answer that first because the rate is what we are looking at. The rate will increase when we exercise because the heart needs to pump a lot of oxygen and glucose to the body cells, all right? Okay, next, this is an important one. Now, you know that every year there will be similarity and differences question. It's found in the structure, it's also found in the essay. Essay definitely have question 9 and 10. And then there will be some similarity and differences question in the structure itself. So this is an example of a similarity and differences question. So for example, they ask for the similarity and differences between tissue M and N. Okay, so the similarity is both muscle tissue, and since they are muscle tissue, they can contract and relax to produce movement. So for M, M is cardiac, N is smooth muscle. So M is found on the heart, wall of the heart, and this is found on the blood vessel in our small intestine. So the heart, the function of the muscle is to produce heartbeat, all right? And this one is to cause peristalsis. And the rate of contraction and relaxation for the heart can be up or down, increase or decrease, but for the blood vessel is usually very slow. Okay, so this is one example of a question 1 in the structure paper. Okay, then another one here would be linked to chapter 9 here. This is chapter 9, so that means if they can link to chapter 10, chapter 2 link to chapter 10, chapter 2 link to chapter 9. So this is villus. So they ask how tissue N helps to carry out its function, how helps small intestine to carry out its function. So tissue N is a smooth muscle, so it can contract and relax to produce movement. And the movement here, you can be more specific because it's actually peristalsis. Now, the reason, the function for it to be able to carry out peristalsis is so that more nutrient can be absorbed. All right. Remember, peristalsis is also a physical digestion. So it helps to absorb more nutrient, right? So that is the function. So this is a human tissue. Okay, let's look at a possible plant tissue. So the plant tissue here will be these 3 plant tissue, either the parenchyma, collenchyma, and sclerenchyma. All 3 tissues are ground tissues. So the similarity that they are ground tissues, all right? They fill up the cortex. So how do you differentiate between these 3 tissue is that the parenchyma are usually rounded. They look like round potato, and they have very thin cell wall. So their function is to store product of photosynthesis. And then you have collenchyma. Collenchyma is polygonal shape with unevenly thickened cell wall. And the cell wall are thickened with hemicellulose and pectin. So this is to provide mechanical support for herbaceous plant. And the last one is sclerenchyma. You can see that the wall is very thick and dark. Okay, sclerenchyma tissue provide mechanical strength and support to bigger, especially plant that have undergo secondary growth. Okay, so these are the 3 types of plant tissue. And please remember, plant tissue, they can link to this zone of cell. Make sure you have the word cell, zone of cell division, zone of cell elongation, and zone of cell differentiation. Okay, either at the shoot tip or the root tip. So make sure you know that what happened, how do you identify the tissue in zone of cell division. The cells there are usually have large nucleus. With the nucleus, usually the chromosome are undergoing different stages of mitosis, such as anaphase, metaphase. Okay, so you can see that here, there's this photo here that shows you the different stages of mitosis in a zone of cell division. So the cells here are meristematic cells, or tissues. Then zone of cell elongation are a zone where you see small vacuole combining to form a large vacuole, right? And then finally, you have zone of cell differentiation. So how do you tell where does the zone of cell differentiation begins? It begins when you see there are root hair cells coming out from the side. And here in the shoot, it will be, you see some xylem and phloem. And also, linked to this chapter, which I think is quite important, is the life cycle of the tree plant, which is the annual, biennial, and perennial. Okay, so that one I leave it to you all to go and revise. Annual, biennial, and perennial, the 3 life cycle of a plant. Okay, and of course you need to know the rest, like for example, the tissues that are found in meristem apex and also lateral meristem, which consists of cork cambium and vascular cambium. You need to know how the lateral meristem cause secondary growth. and how the apical meristem cause primary growth. Okay, I just state only, you all have to go and read up a bit about primary growth and secondary growth, right? So because there's so much I have to cover. Okay, let's move on to chapter 4. Okay, I'm skipping some chapters, not because it's not important, but because I don't have time to cover everything. So I'm only picking up, focusing on things that I think is very important, right? So the next one is, of course, nucleic acid. Now, in nucleic acid, you know there's DNA and RNA, so make sure you guys know how to draw lah. You see, this is 2 polynucleotide here. So the 2 polynucleotide here, you make sure you know how to draw the, you see this one, this is phosphate, right? Okay, wait. Okay, this is phosphate group. This is the pentose sugar. Make sure it's drawn pentose shape, and then you have the nitrogenous base. If possible, please draw it complementary. That means the shape must match. And then this is one polynucleotide, and then this is another polynucleotide, but you can see that the pentose sugar is upside down. So just count 1, 2, 3, and then you have one nitrogenous base coming out, and then another 1, 2, 3, another phosphate group come out. So practice drawing. You know that drawing will come out. I don't know which drawing will come out this year. Last year came out root cells. Last year came out auxin. So DNA can be one possible drawing that might come out, besides the one on the zone of cell elongation just now, where you need to draw the vacuole very long and large. Okay, so if this one come out, obviously it's quite easy to draw. But if this one come out, you make sure you know how to draw correctly with the polynucleotide this side upside down. Okay. Okay, so practice drawing, yeah. Okay, then they would like to ask about function of nucleic acid. So this is the function of nucleic acid. Okay, by the way, the P is hydrogen bond. And then nucleic acid, of course, carry genetic information which determine the characteristic of the phenotype of an organism. Okay, moving on. This is another question they may link to talking about the synthesis of protein from DNA. So make sure you use 2 words here, which is transcription. So from here, DNA to mRNA is transcription. TRANS, transcription, yeah. And here is translation. Okay, so remember that the DNA here have the bases which will be transcribed to mRNA. So every 3 bases that is transcribed is called a codon, and then tRNA will bring the amino acid. Okay, and the tRNA has the anticodon which pair with the codon on the mRNA. So the anticodon of tRNA will pair with the mRNA. So you get the sequence of amino acid being linked up to form a polypeptide. So what is important here, looking at this question is probably 3 marks. So you must use the word transcription and translation. So make sure the bases you say transcribe to the base on the mRNA, and then finally you say that the bases will then end up being translated to a sequence of amino acid that will link up to form a polypeptide. Okay, so this is important. And of course, if without transcription, then therefore there won't be any synthesis of protein. Okay, next stop is the second comparison. Now, earlier I gave you 1 comparison. In fact, I gave you 2, which is the muscle as well as the ground tissues. Okay, so now this is another important one. The differences between DNA and RNA. So this is the similarity and these are the differences. Okay, I won't elaborate now. Let's move on. Okay, so, and then the third drawing, which I want you all to pay attention is this. Make sure you all know how to draw the triglyceride, linking the tail to the glycerol. And how do you describe this process? Make sure you use the word 1-3-1-3. That means 1 molecule of glycerol combined with 3 molecules of fatty acids condenses to form 1 molecule of triglyceride and 3 molecules of water. Okay. The rest, like maltose plus maltose, you get, glucose plus glucose, you get maltose. The rest of the monosaccharide, which combine to form disaccharide, that one you all go and revise yourself, yeah? The 3 types of monosaccharide that combine to form disaccharide, okay? All right, moving on, we reach now chapter 5. Okay, chapter 5, make sure you know the common term that we use to describe about the reaction between enzyme and substrate. For example, complement, the word complement, the substrate complement to the active site of the enzyme to form a complex enzyme substrate before become enzyme and product. Okay, so here is the third drawing that you need to know. Okay, the third drawing is this. If they come out with an enzyme, let's say it's the shape of E, yeah. Okay, make sure, let's say this is the product. Oh, sorry, this is the substrate. So the substrate will bind to the active site, right? So you'll get a complex enzyme substrate, right? So make sure you don't have a double line. Just make sure this one goes into here. And then after that, it breaks up into the enzyme back to its original shape and size. Okay, make sure you retain the size of the enzyme. The enzyme does not change shape, does not change size after the reaction. So you draw exactly the same as the earlier picture. Okay, so drawing of enzyme substrate may come out. I'm not sure. I'm just telling you all. So to pay attention on how to draw. And the word like complement, the substrate fixed to the active site, okay, is complement to the active site, right? And then another thing you need to know is like there are 4 factors that affect enzyme activity, right? Temperature, pH, concentration of substrate, and concentration of enzyme. Now, make sure if it's high temperature, you use the word denaturation. If it's the temperature just about to rise, then you say effective collision between substrate and enzyme. And then also words like you have to know that the rate of reaction is maximum, right? Especially when is here you have concentration of substrate, let's say, and then here you will reach maximum rate here. Here the rate of reaction, this is rate of reaction. So you reach a maximum rate here, and then it will plateau off. That's because the concentration of the enzyme become the limiting factor. So make sure you know all these words lah. Limiting factor, denaturation, complement. Okay, don't use your own language. And then of course, you need to know like the word optimum. Make sure you say optimum temperature. Let's say 37 is optimum temperature. Use the word optimum. Okay, so that is the first few basic questions they will ask related to enzyme. So I'm just highlighting one very important aspect of enzyme which they have not asked before, which is immobilized enzyme. Now, immobilized enzyme is enzyme you combine, the enzyme is combined with the inert or insoluble material so that the enzyme is fixed onto the substance. In this case here is alginate beads. So you can see this is alginate beads, which is the insoluble material. In this way, the enzyme will not move. It's immobilized there. So when you pour in the cow's milk, which has lactose, you want to produce lactose-free milk. So the enzyme there, lactase, will hydrolyze lactose into glucose and galactose. So you'll get lactose-free milk immediately. So what's the benefit or advantages of using this immobilized enzyme is that the enzyme is more resistant to increased temperature or pH change. They won't get damaged or destroyed by the change in pH and temperature. And then, of course, they will remain in the same position throughout the reaction. And then after the reaction, you just have to distill out the lactose-free milk without having to separate it from the enzyme. That means you save costs, right? You save costs in not having to remove the enzyme, and the enzyme can be reusable repeatedly to hydrolyze the substrate, okay? And then the other example of immobilized enzyme is, of course, the production of fruit juices, where you put two enzymes inside, pectinase and cellulase. Pectinase is to break down the pectin, which is the protein, and the cellulase is to break down the cellulose. So in that way, you get clearer apple juice, more in quantity and clearer without the pulp. Okay, so chapter 5 just focus a bit on immobilized enzyme. Okay, so many kind of enzyme can be immobilized, right? Okay, next stop is chapter 6. This is chapter 6. Now, chapter 6, the usual one is non-disjunction. I'm sure you all know the non-disjunction, which is homologous chromosome. Like, for example, they ask non-disjunction, so Down syndrome, right? So Down syndrome is caused by non-disjunction. And you must say during anaphase 1. If you say homologous chromosome fail to separate, then you must say anaphase 1. If you choose to say chromosome only, then just anaphase 2. But I suggest you use 1 only, which is anaphase 1. Then you say non-disjunction occur on chromosome number 21. Homologous chromosome number 21 fail to separate during anaphase 1. Okay, so what you get is actually one gamete that has 24 chromosome, and the other gamete is 22 chromosome. So the 24 chromosome gamete will fertilize the 23 one, then you get a zygote that has 47 chromosome. Okay, so that is trisomy 21. Right, so the next possible drawing that they may ask you to draw is this. So this is a non-disjunction process here is happening here. So they want you to draw the outcome in anaphase 2. So you see here got 3, here got 1. So in the end, you get like this. Anaphase 2, so you get like this. So this one end up with an extra chromosome, this one minus 1 chromosome. Okay, so this is the next drawing that you have to pay attention. Okay, so moving on, actually, other than that is you have other type of chromosome drawing like this. Like this is mitosis, this is meiosis. Make sure you know how to identify them, and what's the difference in terms of their arrangement. Mitosis metaphase with metaphase 1 of meiosis. So make sure you use the word homologous chromosome arranged at the equatorial plane. The other one, chromosome are arranged at the equatorial plane. Okay, so the drawing is very important. And then let's say they ask you, how do you draw the stage after P? So let's say this is P, right? P is mitosis, metaphase, right? So after metaphase is anaphase. How do you draw anaphase? Okay, this is another drawing. Take note. This is how you draw. Okay, so make sure the centromere is pulled by the spindle fiber to the centriole, not the other way around. So the drawing must with the centromere towards the direction of the centriole. Okay, so label them correctly. Okay, okay, so this is another possible question. Okay, this is the one that is the Down syndrome one. Right. Okay, so moving on now, we move to chapter 7. Now, chapter 7 is actually a very important chapter this year. Okay, so I make sure you're focus on chapter 7. The chapter is a very short chapter. You can study really, really study hard about chapter 7. Okay, there are many questions they can ask from chapter 7, but the basic is this. If you can tell the three formula that is found in chapter 7, you can answer any question related to chapter 7. Okay, so for example, this one, let's look at this equation. Now, the common mistake made by a student is that, the process they always say oxidize. This is not oxidation. Just say glucose breakdown into 2 molecules of pyruvate. Just use the word breakdown. Because then after that, this is in cytoplasm, right? Then after that, only the pyruvate will enter into the mitochondria. Then only you say the pyruvate is oxidized into carbon dioxide, water, and energy. So this is a aerobic respiration, which you need to know the formula, of course. And this is the second one. Okay, this is 2898, 2898 kJ of energy. Alcohol fermentation, make sure you know, is glucose to ethanol carbon dioxide energy. So this is 210 kJ. This is lactic acid, glucose, lactic acid plus energy. Remember, there's no carbon dioxide. And there's no water. There's no water in alcohol fermentation and glucose. Unlike here, you see there's water here. Okay, and then there's no carbon dioxide here. So the energy here is 150 kJ. So there are only three number you need to know. I'm sure you all can memorize just three number: 2898 kJ, 210 kJ, and 150 kJ. Okay, it helps because it will help you to later talk about the differences. Now, what kind of related question they will ask in chapter 7? Now, number 1 is milk fermentation. Now, milk fermentation, you must remember, is lactobacillus that will act on the lactose, break it down to lactic acid. Then the lactic acid will coagulate the casein and turn it into yogurt. All right. So it is the lactic acid that causes the sourness in yogurt. Or the other possible question is about paddy being waterlogged all the time, and the condition is always insufficient oxygen or lack of oxygen. So the paddy will carry out alcohol fermentation. Now, paddy is able to do that because they have the enzyme alcohol dehydrogenase that can break down the ethanol molecule, which is actually toxic, into carbon dioxide. Okay, so they can survive even though they are always submerged underwater. However, if you talk about like Banjay now, right, a lot of area of Banjay, and then you have all these normal tree submerged under Banjay water, the most they can survive is 1 week. They will die because they don't have alcohol dehydrogenase to break down the ethanol, so they cannot survive. Okay, so these are two possible questions related to chapter 7. And of course, the most important is next is another differences question. Now, this is the third one. You say this can come out in essay. I don't know. If it comes out in essay, you probably have more differences that you need to write. But if it comes out in structure, you probably need to know only 2 differences. Okay, so these are the similarity. Now, I don't have to elaborate all this. It's just that I want to highlight to you. Here, you have the common word that students don't use is the complete and incomplete oxidation of glucose. Okay, complete, incomplete, occur in the presence of oxygen, limited oxygen, on absent. Occur in cytoplasm and mitochondria. This is cytoplasm, produces water, does not. And then the product and the energy. Okay, now if let's say instead of lactic acid fermentation comes out, then you have this as my sample answer that I give you. Now, what I didn't prepare for you is aerobic respiration with ethanol fermentation. But actually, you can do it yourself very easily because P2, P3, P4, P5 is almost the same. It's the same actually, only P6 and P7 change. Okay, so go and do some homework, go and do a differences question, aerobic with alcohol or ethanol fermentation. Okay, P2, 3, 4, 5 is the same for the ethanol fermentation. Okay, now what else will they ask? What kind of question? This is another possible one. This is about a person exercising. Okay, so before lactic acid is low, the concentration of lactic acid is low. After or during will be high. After will take some time for the lactic acid to be fully oxidized to become pyruvate or to become food, CO2 and energy. So it takes time for the recovery. So the red color here is oxygen depth. The Q section here is oxygen depth repaid after you stop exercising. Okay. Okay, okay, look at this question here. Now, a runner runs very fast after an intensive training. Explain why this is possible. Okay, why is it important for them to be consistent with their training schedule? Now, the more you exercise, the more mitochondria you generate in your muscle cell or muscle tissue. So your cellular respiration become more efficient. so you can oxidize glucose more efficient to produce energy. Okay, that's the reason why training is very important. And then for this question here, the person went into oxygen depth. Okay, so what will happen if we start from a distance of 200 meters of the race? So the muscle will undergo oxygen deficiency and then undergo lactic acid fermentation. So your breakdown of glucose will be incomplete, right? Okay, so after finishing the race, the athlete rests and undergo recovery. State the importance of this in recovery. Now, recovery is very important because recovery, you need to oxidize the lactic acid. Otherwise, your cramp will not go away. So you after the lactic acid is fully oxidized, then only you say oxygen depth is big. Okay, this is another question here where Paddy, the one that is submerged is actually ethanol fermentation. The one on top is actually aerobic respiration. Okay. All right. So now we are done with chapter 7. Moving on to chapter 9 a bit. Okay, chapter 9. I'm just going to scroll very fast. This is about the function of liver. What does liver do? Liver synthesize plasma more protein, convert excess amino acid into urea to be excreted out through the kidney. So liver also with the insulin help to convert glucose, excess glucose into glycogen, right? And then when it's insufficient, the glucagon will convert glycogen into glucose. Okay. Okay. Now this is suku-suku separuh or quarter half question that might come out in essay. So if let's say they ask you to meal, sajian, eh? So sajian means you got to propose what kind of meal is suitable for this group of specific person who have specific condition like cardiovascular problem, obesity, and cancer. Okay, so refer to the quarter half. Please refer to quarter half. Quarter here means quarter of protein. So you are suggesting for a heart problem patient, you cannot suggest eat red meat. You must say protein like chicken, white meat, or fish or salmon. The way you cook, how? Steam it, boil, okay? Not fry, okay? Because he has heart problem already. So suggest the way, the method of cooking that will not increase cholesterol in his body, in his cell, okay? So grilled salmon because it has high omega-3. So when you state something, you must justify, right? Say, for example, you can suggest the quarter carbohydrate, oat, cereal, brown rice, because it has less starch. So it also has able like oats and cereal to reduce cholesterol. And the other half is, of course, vegetable and fruits. Okay, you can say fiber to help with digestion, vitamin is to maintain good health. The same goes for obese people, brown rice, roast chicken, don't fry chicken. Obese people maybe have to reduce their sugar intake, so take more vegetable, leafy vegetable, dark green vegetable. Okay, less fruit, less sweet fruit like grapes. Avoid all those that will increase your glycemic index. So obese is almost the same as diabetic. And then of course, cancer patient, you always talk about you want to help the patient to recover faster. Protein is very important. Again, do not suggest red meat. Suggest lots of fruits and vegetables because they will increase their immunity. Okay, so cancer patient, their immunity can be quite low. Okay, so this is a quarter half. Now I'm going in now to some possible KBAT question related to chapter 9. We are now in chapter 9. Okay, for example, they say, the patient have to remove the pancreas. So what will happen? Pancreas is very important. Pancreas is exocrine and endocrine gland. Both they function in two different systems. So remember, they secrete lard, which is lipase. Okay, A stand for pancreatic amylase and T stand for trypsin. So without or half the pancreas removed, then you will say there will be less lipase to hydrolyze lipid into fatty acid and glycerol, less trypsin to hydrolyze protein into protein is not protein, right? Polypeptide to peptide. And then lastly is less pancreatic amylase to hydrolyze starch into maltose. So, which means that what must the person do? Eat less, less protein, less lipid, less carbohydrate. Okay, that is for the function as a exocrine gland. Now, for endocrine gland, now you know they secrete two very important hormones, which is insulin and glucagon. Now, if you look at a pancreatic patient, they will have problem in controlling their blood glucose. So when the blood glucose is high, they have insufficient insulin. So they cannot convert the excess, they don't even have excess if they are not eating sufficient glucose. So the glucose cannot be converted into glycogen. And then if they have excess also cannot. And then at the same time, their glycogen cannot be converted into glucose because lack of glucagon. So they cannot control their blood glucose. Okay, so that is the effect for a patient who has to remove partial of their pancreas due to cancer. Okay. So what can the person do? Reduce the intake of all the three food I mentioned just now. Possibly have to go for insulin, glucagon injection, and possibly have to go for pancreatic organ transplant. Okay. Okay. So we're going to look at some other related question with chapter 9. So this is the labeling of mucus, chief cell, and parietal. So make sure you know all their function, especially the difference pH between the mouth and the duodenum. Okay, the medium of reaction. Gallstone, what's the effect? Gallstone is the accumulation of fats and cholesterol, so the bile cannot flow out, cannot channel out to the duodenum. So therefore, the lipid cannot be emulsified, and the person cannot eat lipid or fats because they cannot digest the fats. Okay. Next is make sure you know this is epithelial cell, capillary, lacteal, and their adaptation. How is the structure P adapted to its function? Make sure you know all these are the total surface area and so on. Okay, and then the other thing is this. Okay, from absorption, make sure you know from digestion it goes to absorption. So make sure you know absorption of glucose and amino acid is absorbed through the epithelial cell and then into the blood capillaries, whereas fatty acid and glycerol need to recondense back again to form tiny droplets of lipid before diffusing into lacteal. Okay. And then remember, this is assimilation from the blood capillary. Glucose and amino acid get transported through hepatic portal vein to the liver. So in the liver, this is where the assimilation process occurs, where the liver will synthesize plasma protein and enzyme, and also excess amino acid converted into urea through the process of deamination. And glucose always used for energy, excess change to glycogen and stored. Okay, so the same thing you can say in the cell. For amino acid here, you can say repair damaged tissues, right? Glucose is the same. Now look here, the lipid does not go to liver, it goes straight to the body cell. That's where the assimilation of lipid occur, where the lipid is used to form adipose tissue and so on. Okay. Now, the excess intake of protein can cause a lot of uric acid. Uric acid can form in the joint, can form crystal in the joint, which leads to gout. So gout can actually become very, very painful, especially at our joints. So you have to avoid food that is contributing to the formation of this crystal uric acid, such as liver, kidney, and sardine. Okay, another possible K-back question is this. The person have, just now I said pancreas, right? Now is stomach. Okay, so if his stomach is surgically removed, then what can you do? What can he do? Okay, so first of all, he has to reduce the intake of protein, right? Because he has less pepsin to convert protein into polypeptide. And milk also he has to avoid because he doesn't have renin to coagulate the milk. And his stomach now is smaller, so he has to eat small frequent meal and small frequent meal. And then he has to eat semi-solid food because it's harder now with a smaller stomach. He has less muscle to undergo peristalsis. And lastly, he has to ensure that the food that he eat is clean and hygienic, okay, with containing, because his hydrochloric acid is less now. Okay, so that is what he has to do. All right, so I think I have done all chapter 9. So I'm skipping chapter 10 because of time. I will just have to tell you all to focus a bit on mechanism of blood clotting, and also the important thing is this, the sequence and the contraction of the heart muscle. The SA note, the AV note, that one I leave it to you all to read up. Okay, and we are moving now to chapter 12. Okay, the eight chapters of Form 4 is very important. 12, 13, and 15. Okay, 12, 13, and 15. So now let's look at chapter 12 here. Chapter 12, comparison between sensory neuron and motor neuron. So this is, there are about 7 differences. Okay, cell body, where is it found? I'm not going to go through. Okay, I think you all know already. This is located at the side, not middle, yeah. And then another thing is synapse. Make sure you know how to explain the process of transmission of nerve impulse through the synapse. K is the mitochondria. Synaptic vesicle, which contain neurotransmitter, and then the impulse coming from one direction coming down will trigger the synaptic vesicle to secrete neurotransmitter. Okay, so the neurotransmitter then will diffuse across the synapse and bind to the receptor on the receiving neuron before a new impulse is generated. So make sure you know the whole process and the effect of drugs, both drugs, stimulant and depressant drugs, on the transmission of the impulse across a synapse. Like depressant slow down the transmission and also block the neurotransmitter to bind to the receptor. And then stimulant, of course, speed up the transmission, and more nerve impulse is being generated. Okay, so you need to know, okay, go and prepare a bit on the effect of drugs as well as alcohol. Okay, alcohol has effect both on nervous system and also endocrine system. For nervous system, a person who take alcohol will not be able to judge distance very well. May lead them to think irrationally a bit, and then of course the coordination, their reflex action is what the most worrisome, because their reflex action is slower. So if they drive and they drink and drive, and then they see an object on the road, they may not brake in time, okay, because their reflex action is slower. And then for endocrine system is the less ADH is secreted, so less water is reabsorbed from the distal convoluted tubule and collecting that into the blood capillary. So that means more urine is produced. So the person who takes alcohol will get dehydrated because they lose too much water. Okay, they pass urine a lot. Myelin sheath, the importance of myelin sheath, go check it out, okay? And then Parkinson disease, Alzheimer disease, this is dopamine, this is acetylcholine, okay? Less of neurotransmitter. This one affect motor neuron, this one affect reasoning, affect the brain actually have deteriorate. So Alzheimer, it deteriorate, so it may affect their reasoning, their memory, so they will have lose their ability to do the basic thing in life lah, like talking, walking. They can't find their direction home. For Parkinson, because it affect the motor neuron, so that's why they will, they tremor as they walk, their hand will shiver like that. Okay. And then of course, voluntary and involuntary action, the difference between voluntary and involuntary. So maybe they'll give you picture of two different action. One person is playing football. Okay, that one of course is voluntary. Then another person who is showing the intestine, just had a meal and then showing peristalsis. So that's involuntary. Okay, so this is the differences. Wait, okay, so that is for chapter 12. Of course, hormone, you're going to read yourself. Hormone, I have no time to tell. Okay, so this is the synapse. Now, I'm going to just touch very briefly chapter 14. So tendon, ligament, similarity, strong, not inelastic, elastic. Okay, make sure you all know the three facts, right? To talk about how you bend your arm. Okay, bicep contract, tricep relax, pulling force produce must be transmitted through the tendon. So radius is on top, ulna is at the bottom. So since it's bicep contracting, so it's transmitted to radius. Okay. And then the drawing when bicep femoris, when it is bending of the leg, you need to draw it to be a bit, the muscle to be a bit sort of fatter a bit. And tricep here is actually the quadricep femoris. You have to draw it thinner and longer. Okay. And then what happens if you injure your knee? Okay. Okay. How to explain from position X to position Y? How do you explain S and R? So in this position, of course, R is contracting. R is bicep femoris. S is quadricep femoris. So from here to here, that means from contract to relax, from relax to contract. Okay. So every time, if let's say you forget about everything, you must remember one thing that will obtain 2 marks. All muscle work antagonistically. They have to work in pair. When one contract, the other one will relax, and vice versa. Okay. And then, of course, joint, the different function of joint, there are differences between ball and socket and hinge joint. Okay, this is the differences between scoliosis and osteoporosis. Okay, and this is osteoarthritis. Remember, the cartilage become thinner, ligament become loose in shorten, and most importantly is the wear and tear of cartilage. Okay, and the decreased amount of synovial fluid. This is practices that an athlete need to do to maintain a healthy musculoskeletal system. There are 4 aspects. Practice good posture. Number 2 is wear proper clothing. Number 3 is carry out warming exercise. And number 4 is diet. Okay, proper diet. Okay, animals, don't forget, animals, this is minor, major. Minor contract up. So if there's any injury of minor cannot contract, therefore the wing cannot be pulled upwards. And then if this one come out, make sure you understand that the similarity is that all animal, regardless, they all have antagonistic muscle. They have a pair of muscle that can contract and relax. Okay. And of course, they have skeletal system. Now, grasshopper have exoskeleton, earthworm have hydrostatic. Exoskeleton support and protect the organ, whereas hydrostatic is actually maintaining a rigid body for the earthworm. Okay. The name of the muscle. Okay, the name of the muscle, circular, longitudinal, and what is it used for? Okay, that is chapter 14. Now, chapter 12, just a little bit additional for chapter 12, reflex section. This is major. Remember, if you are to ask to draw the neuron that is found here, this is sensory neuron going in, okay, going in through the dorsal root, and then the motor neuron at wait, I think I got it wrong. The other way around, sorry. Okay, so this is, they label this is sensory neuron. So this one go in, this one come out. So sensory neuron is to transmit nerve impulse from the stretch receptor here to the spinal cord here. Now make sure you draw like that with a cup without relay neuron, straight away motor neuron, because this is the simplest pathway for our neural pathway. No relay neuron, only got two neuron, sensory neuron and motor neuron. Okay. So when you hit the tendon, the stretch receptor will be stimulated to produce a nerve impulse, which then transmit to the motor neuron from sensory to motor. And then the motor go to the effector, which is the quadricep muscle, and then your knee jerk. Okay, so we are now chapter 13. Now, chapter 13, okay, is important. I've done with chapter 14. Go back to chapter 13. Sorry, should have gone 13 before 14. Okay, 13, diabetes. I'm sure you all know about diabetes. Now, more importantly, I want to focus is this. Chapter 13 is this. This fella here is very important, nephron. Make sure you know all the labeling. How is urine formed? Okay, I don't have time to explain. Ultrafiltration, reabsorption, and secretion. So remember, if you talk about reabsorption or you talk about secretion, make sure you talk about the direction. So you talk about reabsorption. So if this is tubule, this is blood capillary. Reabsorption is from tubule to blood capillary. If you talk about secretion, then secretion is from where? From blood capillary to tubule. So it's opposite direction. So you must know, you must tell this. When you cannot just say reabsorb, you must tell reabsorb from where to where. Okay, so if you understand the urine formation, then you can understand all this. Why this one 0 at structure Y. Okay, this is loop of Henle. So you should be able to explain this. As make sure you also know how to explain ultrafiltration. Okay, how does it happen here? How does ultrafiltration occur? Okay, and this is what? Okay, this is BOP high, BOP low, BOP high, BOP low. Okay, BOP high is when you take a lot of salt. BOP low is when you take a lot of water, drink a lot of water. So focus on one, the other one is just the opposite. Okay, BOP high, blood osmotic pressure high, salt concentration is high, osmoreceptor is more stimulated. So I always ask students to choose the BOP high because BOP high will talk about osmoreceptor more stimulated. So you can remember better. Pituitary gland also more stimulated, more ADH is secreted. Therefore, more water will be reabsorbed. So this is where I meant you have to tell from where to where. Okay, at the same time, adrenal gland is involved in the reabsorption of salt. So you already BOP high, you don't have to reabsorb so much salt. So your adrenal gland will be less stimulated, less aldosterone is secreted. So less salt is reabsorbed, okay? The outcome is, of course, the urine become more concentrated and the volume is less, and therefore returning the BOP back to normal. Okay, the other one, BOP low, is just the opposite of BOP high. Yeah? Okay, so one regulation question will come out. I don't know which regulation question will come out. So chapter 13, you have temperature control, glucose, blood pressure, right? Carbon dioxide, and BOP. There are 5 regulation. So I only have time to talk about BOP and also blood pressure. Okay, so the other 3 you got to go and read yourself because regulation will come out, right? So this is blood pressure. Blood pressure when running is of course high. When a person bleed is of course low. So if you focus on one, like how I teach you earlier, BOP, so if you focus on the high, then the low one will be just directly opposite. So BOP high, okay, more stimulated. Okay, baroreceptor will be more stimulated, then will send more impulse to the cardiovascular center. So vasodilation occur. Okay, so vasodilation occur means the blood capillary, the blood vessel all will be, enlarged a bit. So it's easier for blood to flow. That will decrease the resistance in blood flow. Okay, so with less resistance of the blood flow, that means your heart don't have to pump so hard. Remember, your blood pressure is high, so you got to make your heart, pump slower. So you need a weaker contraction of the heart. If you have a strong contraction of the heart, then BOP is going to shoot up even higher. So you need to weaken the heart contraction. So the outcome of the blood vessel being dilating means more blood can flow, less resistant. Okay, by the way, dilation and constriction is for blood vessel. Contraction and relaxation is for muscle. Don't use wrongly. Okay, the opposite is true. So I got no time to talk about the opposite. Okay, moving on. Okay, a little bit on chapter 14 here. This is minor, major. Okay, I just want to show you fish here. Make sure, just now I talked about insect, I talked about the bird, now is fish. Fish is myotum muscle, shape W. Okay, so the myotum muscle is placed, if you look from top, this is the fish. So they have the vertebral column here. When the right side contract, the left side will relax. So this one relax. Right contract means the tail will move to the right and vice versa. Okay, so that's how they move and whip their tail. Okay, I'm reaching chapter 15. Okay, it's 11 o'clock. Okay, I haven't finished from 4. Okay, chapter 15, this is very important. Chapter 15 also is another chapter you have to study well. This is a question about the relationship between hormone, follicle, and endometrium. So you need to know how from the beginning about FSH, LH, estrogen, how they all related. Okay, so and how they stimulate the follicle grow, and then estrogen come out, more endometrium will become thicker and so on. So the whole process you need to know. Okay, the cycle. Okay, I have no time to go through. You all have to go through yourself. And then the function of HCG hormone. HCG is it's like LH, how it will stimulate ovulation in a person who cannot ovulate. Okay. And then this one. Okay, this one is spermatogenesis and oogenesis. Okay, the differences. This is the differences question. This one you got to read. If you don't read, right, this is pretty hard to remember. Okay, so you have to understand spermatogenesis first and oogenesis. Then only you can tell the differences. Okay, so I have no time to go through. And twins, there's a lot of similarity and differences question in chapter 15. So twins is another one. This is identical twin. This is one is identical twin, one is non-identical or fraternal twin. Now you need to know their differences lah. Okay, and finally this one. This is the differences between sigmoid curve and also the insect, which is a step-shaped curve. Okay, you need to know their differences, right? So ecdysis, you need to be able to explain how ecdysis occur in insects like grasshopper. Why do they have to undergo ecdysis? Why can't they grow like normal? It's because of their what? Their exoskeleton is very hard. So in order to grow, they need to remove their exoskeleton first. The exoskeleton is very hard and rigid, so it limits their growth. So they have to shed off their exoskeleton, come up with a new exoskeleton in order for them to grow. So you have to understand and explain the process of ecdysis and of course the differences between both. Okay, that's all for Form 4 section. Now is 11:03. I open up now for Q&A for a while for Form 4 section only. Okay, come on. Any question you can type in the chat box. Okay. Anyone would like to ask any question? Anyone? Essay can use table. If it's comparison, can. If it's not comparison, question cannot. Comparison got similarity and differences, right? Similarity cannot write in the table, only the differences. Point form, of course, must be logical lah. Your teacher say is correct. You use so short form, no one understand except you. Of course, cannot lah. Like for example, right? For example, okay, I give you one example now. You say insect, human. You didn't even say occur. You don't have the word occur. You just say insect, human. Who knows what you're talking about? Insect, human. What is insect, human? Okay, or you say chitin, calcium. You don't even say skeleton is made out of chitin. So you cannot be so short form until no one understand except you. So you got to short form like this can, skeleton made out of chitin, skeleton made out of calcium. Yeah, I just said that we can use table to bandingkan. Yes, in essay. Yes, correct. For drawing, at least must have one label, right? Label lah all. Why label only one? If you can label all, label all. Why label one? Okay, and make sure there's no arrowhead. No arrowhead. Labeling got no arrowhead. Don't have arrowhead. Cannot have arrowhead. Before what? Sorry, Oh, okay. You mean what? Sorry. Yeah, you can. That is how I write what? Occur in human. Can. Just don't write human and insect. Okay, this is point form lah ni. Ni point form lah. Okay, wait so many. Okay, I've got to scroll back because I might miss a few questions. Okay, wait. Yeah, if you answer in table, definitely it makes life so much easier for the examiner to mark. They don't have to search for your answers. And it's more organized and you don't repeat. Yeah, correct. Okay. Any more? Wait. Omega, now the omega-3 reduces the bad cholesterol lah. How do you improve my KBAT lah? There's no shortcut KBAT in bio. If you have been consistent in your work, you listen to teacher, KBAT has to be a spin-off from what you already know. It cannot be somewhere drop down from the sky and suddenly out of syllabus question. It will not be like that. It will be related to the syllabus. So they will not ask something beyond your syllabus. Okay, so how do you improve KBAT is you take from the core question and then think what is it related to, which topic is it related to. Okay, and then link to the basic related to the KBAT question. That's how you answer. Okay. Any more? Structure, can I use table by drawing the frame? Okay, most of the time now, they will give you a table in the structure. Okay, say for example, they say here X, this is structure, this is structure, this is X, this is Y. They give you 2 row, you don't pandai add row, cannot add row. Additional of row, even though it's correct comparison, no marks. Okay, say for example, this row correct, okay, 1 mark. This row salah, comparison salah. This row betul. Okay, examiner will not take first and third. They look at first and second because that is what provided. So any additional row will not be counted, even though you're correct and the second row is wrong. We will not take 1 and 3. And then another thing is first attempt only. That means you know, a lot of students they are very unsure one, no. So they will say maybe green green, don't use the word and if you are not sure, green and blue. Now, when you write like this, right, wrong, cancel, right. This is correct. This is right. This is wrong. But you use and ma, so wrong, cancel, right. So 0 mark. Although green is correct. So if you have one answer, write one answer. Don't and okay. If you think you're not sure, use the word or. You use or, right? You didn't say and, then possibly your teacher will choose this. The correct answer for you, which you wrote on this after the first one. But try to limit to one answer if they say one answer lah. Okay. And first attempt only, first attempt only. And then of course spelling. They are very strict now. I'm sorry. They are very, very strict with spelling. Make sure it's correct spelling. Okay, if you want to add frame in a line, let's say this is structure, right? They ask for comparison question, but then they give line. Yes, you can. Yes, you can. But still fall within the line given. Don't draw extra line and then form another row. Cannot. If they give you 2 lines, then you draw the 2 line, a frame within the 2 line. Yes, can. Okay, next. Okay, so spelling error is 0. They are very strict. I'm so sorry because there's so many students simply spell. So your next 1 hour, just spend time. Don't assume you know the spelling. Okay, don't assume. Bruguera, how you spell Bruguera? Even photosynthesis student also answer spell wrongly. Last year, you know how many students spell wrongly meiosis or not? So many students spell meiosis wrongly in the essay. So if let's say you write a very nice sentence, memang correct one, but meiosis spell wrongly, O-I-E or whatever lah, this is wrong spelling. Wrong. The statement correct, spelling wrong. Okay, next, wait lah. Okay, any more? Wait. Form 5 topic come out more. No, there's no such thing as Form 5 topic come out more than Form 4. Equal. That's why I give 1 hour each to each form. Will there be anything happen if I write my answer over the answer line given? Will I be penalized mark over the answer given? You mean structure or are you talking about essay? It's just don't write over the answer if it's stable lah for structure. I hope I did not miss any question. The drawing enzyme need to use ruler to maintain same. Well, you can to help you to maintain. Kebanyakan soalan suruh menyatakan kesan negatif kan cikgu. Tak semestinya. Kalau dia tanya dalam soalan kesan negatif, you jawablah secara negatif. Contoh, sekiranya tiada jasad Golgi, apakah yang akan berlaku kepada sintesis protein? Let's say they ask a random question. Then you say, tanpa jasad Golgi, without jasad Golgi apparatus, comma, whatever happened. You have to answer negatively lah. Do we need to emphasize the number of hydrogen bond between nucleotide connection? No need. It's not in your syllabus actually. Okay. How much to improve the timing? I don't get that question. Okay, I think that's all good. Point form not allowed in entire paper. Write full lah. Guys, if your point form, right, honestly, teacher don't know what you meant by your point form. I don't know how singkat is your singkat. Okay, I think that's all. So I have to start my Form 5 now. Okay, any other question, we will try to talk about it in later on. Okay, I have less than 40 minutes for Form 5. Chapter 1, Form 5, make sure you know how to differentiate between these, right? Meristematic tissue. This one I got touched just now, apical and lateral, and their effect of growth. Okay, so their effect of growth. Next stop, this one I already mentioned. Okay, this is the one that I mentioned earlier, the type of annual plant, biennial plant, and also the perennial plant. Make sure you know this one, the decrease in the mass is due to the store food being used up for germination, and then only exponential because here you have the leaf coming out already, so they can carry out photosynthesis, so they can grow very fast, so their dry mass increase. So this is first year is to grow vegetative structure, which is leaf, stem, and root. So the first year. Second year is to produce flower, fruits, and seed before dying. This is annual perennial plant. Okay, so they consist of a combination of many sigmoid curve. So each sigmoid curve represent 1 year growth. Okay, next, chapter 2. This is the difference between palisade and mesophyll, spongy mesophyll. And then differences between light dependent and light independent. Okay, make sure you know the whole process of light dependent. Okay, the whole process of light dependent. The electron get excited and the H2O, the photolysis. Okay, so then continue with the story so that you know how to explain the whole process of light dependent. Okay, next stop, compensation point. Okay, compensation point is important, right? Make sure you know. Compensation point is light intensity. Light intensity when P is equal to R. When P is equal to R is usually during dawn and dusk, or during very hari mendung. You know what's hari? Cloudy day, cannot see the sun. Haze also can. During haze, also P equal to R, certain time of the day, you find that the light cannot penetrate through the clouds. The sunlight cannot penetrate through the clouds. So the light intensity is very low. So possibly the plant is at P equal to R. Now, when P is equal to R, let's say during haze, it's very bad for the economy of the country, because haze is actually water vapor plus the carbon particles, right, combined, forming haze. Carbon particle comes from maybe open burning, and okay, so you get haze, and haze got very bad effect. Number one, it deposit on the leaf, so therefore light penetration into the leaf reduce. Moreover, they get suspended in the atmosphere, so they reduce light intensity. And the third one is the stoma get blocked. So that means the exchange of gases between the atmosphere and the inside of the leaf also reduce, right? So all in all, okay, always remember the rate of, use the word rate of photosynthesis reduce, therefore yield of the plant also reduce, okay? So during haze, most likely plant is at compensation point. So when plant is at compensation point, there's no net gain of glucose. So how is the plant going to grow? So every time when there's a haze that a country experience, maybe for 1 month, the outcome, the effect is very, very bad, because that means 1 month, no yield. The yield of the plant, it doesn't produce fruit, it doesn't produce flower. Okay, so it's very bad for the economy. Okay, above compensation point is P more than R. Of course, this is good, means photosynthesis is more than respiration. So the production of glucose is more than the consumption of glucose by respiration. So the excess glucose produced then can be used to form flowers, fruits, and so on. So definitely P has to be more than R, because there's a net gain of glucose, and consumption of CO2 is higher. Whereas P less than R is in this region N here. It usually happens in the night. Okay, here the net loss in glucose. That means respiration use up more glucose than photosynthesis can produce. Okay, so make sure you all know the difference. For a shaded plant, a plant that live under shades don't require so much sunlight, therefore their compensation point will be lower. Okay. Okay, next stop is this one, transpiration guttation. This one may come out, I don't know. Okay, so make sure you know the differences between transpiration and guttation. Okay, I cannot elaborate. Next is chapter 4. Now, chapter 4, xylem and phloem. Okay, comparison between xylem and phloem. How is xylem adapted to its function? Got two, right? First, xylem is a dead cell. Therefore, the xylem vessel is hollow. So it's hollow, and then on top of that, the cells are connected end to end. So they form a long continuous tube, which allow continuous flow of water. So that is the function of xylem, right? So, and the second thing is the thickening by lignin. So their cell wall is thickened by lignin. Okay, so that add mechanical support, give mechanical support to the xylem so that it does not collapse under the suction pull of the transpiration. So that is the adaptation for xylem. Make sure you know the differences between xylem and phloem. Now, phloem, how do you identify? Very easy. It looks like a vessel also, but except that it has what you call this sieve plate. So you will see sieve plate in between. Okay, sieve plate, another sieve plate, and then there's definitely a companion cell next to it. Okay, that is how you identify phloem. And then for xylem, usually they will have different pattern forming because of the different thickening by the lignin. So sometimes you can see this type of pattern. Then that is definitely lignin. Xylem. Okay. Next stop is this one, phytoremediation. This is chapter 4. Phytoremediation is a method where you use plant to treat water that is polluted. Okay. So the advantages is, of course, they grow very fast. Their roots can absorb all the heavy metal. But the disadvantages is that they take a long time for them to recover the water pollution to turn it into clear water. Okay. Okay, I'm skipping chapter 5 and 6, not because it's not important, it's I don't have time. Okay, so I'm going into chapter 7 now. Chapter 7 is the differences between xerophyte and also hydrophyte. Okay, the difference. This one slash succulent stem, succulent stem, depending on the type of xerophyte. If you are talking about aloe vera, which is an agave, their leaf is succulent. But if you're talking about cactus, then their stem is succulent. So it depends. Okay, this is adaptation for hydrophytes. Okay, this is adaptation for hydrophyte. And the type of root system for xerophyte. Okay, two only. Either they grow widely or they grow very long, penetrating right deep into the soil to absorb water. Okay. Okay, moving on, chapter 8. Now, chapter 8, saprophyte is very popular. Saprophytes are either fungi or bacteria. Both can be saprophyte. They carry out saprophytism, which is to hydrolyze. They digest food outside their cell, okay, in the substratum. Okay, they secrete enzyme outside, and then after that, digestion takes place outside their body or their cell, and then they absorb the nutrient after that. Okay. Now, without them, then ammonification cannot occur. So that means the soil will become less fertile. Less fertile means plant will absorb less nutrient, right? So ammonification is related to nitrogen cycle. So make sure you know nitrogen cycle very well. every step of nitrogen cycle you need to know lah, and the name of the bacteria. Okay, so let's say if this one don't have, what will happen? If this one don't have, what will happen if this bacteria don't have? So you need to know the whole process first. Okay, so this is where the saprophyte is. They digest all the dead organism. So their function is here. See, without process P, ammonification, then you cannot get nitrite. So their contribution to the whole nutrient cycle is very important. All right, the ecosystem will be disrupted. Okay, this is differences, can be differences between a fungi with a protozoa or a fungi with a bacteria. Now, bacteria are all prokaryote. They lack membrane, their organelle lack membrane bound. They are not they don't have membrane, so therefore they are called prokaryotes. The rest are eukaryote. Just remember, bacteria have cell wall made of bacterioglycan. Fungi have cell wall made of chitin. Algae have cell wall made of cellulose, and there are differences. Okay, and the differences between, let's say, algae with bacteria and fungi with bacteria, that you go and do the comparison. Okay. Okay. This is a nitrogen cycle, okay, and they like to ask, why do farmers have to adopt this rotational crop system? Rotational crop system means to plant at least in one of the cycle in 1 year, you must plant one leguminous plant. Now, the leguminous plant, the nodule of their roots contain rhizobium species which can convert the nitrogen in the atmosphere into ammonium compound, correct? So therefore, you need to introduce this plant inside your cycle, 1 year cycle of planting any kind of crops, because you want to enrich the soil with nitrates, okay? Okay, chapter 9. Now, chapter 9, mangrove. Mangrove can come out in question 10 or 11. Okay, now I'm zooming towards the end of the question that will come up, towards the essay question, yeah? So problem faced by mangrove trees. There are about 5 problems. Go figure out all how they adapt to live in their environment. The mangrove have a lot of problem, right? High salinity, high transpiration rate, the anaerobic condition of the soil. How do they overcome each of the problem? Okay. And then this viviparous seedling, why they have viviparous seedling? Okay. And then make sure you know their, wait, okay, their zonation. This is their zonation. Make sure you know their position. Where are they located? Okay, and then this is side view. This is transect view. Avicennia, Sonaratia, their root system, Rhizophora and Bruguera, and how succession occur. So in order to know succession, you need to know what? You need to know their root system, right? Okay, wait. Okay, these are their root system. You need to know their name of their root that belong to each species. Okay, so you memorize their root system. Why? Because then you can explain the succession process. If not, you cannot explain. Because you need to talk about their root system and how the roots will trap all the silt and then to cause sedimentation, resulting in the soil being higher and drier, and then no longer suitable for itself, but more suitable for the next species to take over its location or the place where it once grew. So they will shift forward. That is called succession. So if you know their root system, you know the name, you already got a lot of marks there. So this whole process here is called succession. Okay, and you will know that the question will be like this. This is year 2025, let's say. Okay, they may ask you to project. They ask you to project in year 2060. How would the zonation of the mangrove swamp be? So you got to make sure that you know they will all shift forward. Okay, they will all shift forward. That means the Avicennia will move forward, the Rhizophora will take the Avicennia part, and then so on and so forth. Bruguera will take the Rhizophora space. So, or they will show you two maps, one before, one after, and then they ask you to explain what happened that caused the different zonation over the years. So if you don't understand succession, okay, if you don't understand this whole process of succession, then you cannot explain. So in order to remember this, you need to know this first. This is very important. Okay, so mangrove, what did I say just now earlier was the first thing is the problem they face, right? The problem they face. Second is succession and colonization. Okay, and thirdly is of course the very important one, which is the importance. Now, the importance of mangrove swamp can come out in question 11 because they can show you a piece of mangrove swamp, and then after that, the next picture, they show you the mangrove trees are all cut down, and then they build resort there. And so they will ask you, what will be the effect of cutting down all the mangrove trees? What will be the impact on the ecosystem there? So if you know the importance of mangrove swamp, then that question is the negative, right? So you will turn all this into negative answer, meaning there's no more mangrove trees to protect the coastal from erosion. There's no more mangrove tree to break the wave or to prevent tsunami. Everything is negative lah. So this is very important. What is the contribution of mangrove trees to the balanced ecosystem? Can be balanced ecosystem, can be how to maintain mangrove trees or mangrove ecosystem so that it is provide sustainable environment. Okay, a sustainable environment here means it can continue to become the feeding ground for all the animals so that it can become a generate economy for fishermen. Fishermen can still catch fish from the that area, which is actually a very important site for fish to grow and seafood. Okay, so importance, yeah. Okay, next stop is other than that, chapter 10. Chapter 10, there are three things you need to know. Number one is this one, green tech, the definition of green tech, and this is an example of the implementing green technology in building. So we call it the Green Building Index. Green tech in building your house or building a building. What facts that or factor we have to think about when we want to build a building that fulfill the Green Building Index? Okay, so green tech. Now, green tech can also be green tech in our daily life, like the eco enzyme, the the foliar fertilizer, the bioenzyme and all that, the one that the housewife use, how we convert compost, how we make compost from discarded food and all. So this is green tech in household. Green tech in school. How do you apply green tech in school would be like, for example, the recycle paper or 5R, recycle, reuse, and then you can also like save energy, all right? Save energy, use LED lights, save water, use paper both sides. Those are green tech in school. Okay, that is green tech. And then another one is the second thing. These are all under green tech. So the second thing in chapter 10 is, this is the question 11, which is they show you a proboscis monkey about to extinct. So they ask you, how are you going to ensure that they do not become extinct? Okay, they are already endangered. They may become extinct. So what are your ways to make sure they don't get extinct? Okay, now looking at the question, you have to look lah. There are three aspects here. One is preservation, the other one is conservation, and the third one is restoration. Okay, beside green tech, this is another possible question. Okay, if preservation, you look for the word gazette. Gazette, reserve forest. Okay, so you maintain it. Forest not disturbed yet, maintain it, keep it that way, so the proboscis monkey there will not be affected. Okay, another way is, let's say the monkey is already affected, people go and chop down trees, they don't have a habitat anymore. So it's either in situ or ex situ. In situ means that you still keep the monkey there, you retain the component of the ecosystem as it is, and hopefully after many years it will grow back. The trees will grow back and the ecosystem will return back to its original state. Of course, you have to prevent people from poaching, from coming in, from building new development. Okay. Okay, the other way is ex situ conservation, meaning you capture all these monkey and put them in a conservatory center. There in the conservatory center, you make sure you feed them, you make them reproduce and increase in number. And then maybe when their original habitat has recovered over the years, you return them back to their original habitat. That is ex situ conservation. Now, restoration is actually reforestation. So keyword is reforestation. So the part of the habitat is destroyed, maybe you have to go there and replant new trees. Reforestation is also another amalan, practices that you can do for restoring a mangrove swamp that the trees are all chopped down already. This can be a gotong royong project in a school where people go and plant, manually the sapling of the mangrove trees. So it's called restoration. Active participation of the public, okay, can be also classified under restoration, not conservation, not preservation. So the gotong royong part is the restoration part, where there's active participation of the public in ensuring the area get restored back to its original condition. Okay, so you are restoring back the damaged ecosystem. Okay, this is the second chapter 10 question that might come out. The third one is the practices that can be carried out to ensure environmental sustainability. Now, environmental sustainability is such a huge, famous in thing now, environmental sustainability, what are the goals that the United Nations try to achieve in the year 2030. There are 17 goals, sustainable goals that they want to achieve. So you don't have to memorize all the 17 goals. Your goal is to memorize the amalan, the practices that will bring towards the environmental sustainability. So I'm just going to highlight only environmental friendly transport. Of course, you have to elaborate yourself. And then number 2, biological control method. Number 3, preservation, conservation, and restoration. Number 4, 5R concept. Number 5, green tech. Okay, so you can see there's a bit of overlap with the earlier thing that I said. Other than that, let's say you can also introduce modern technology in farming, in agriculture, using drone to fertilize the plant. Okay, this all will contribute to environmental sustainability because you are not using chemical. Biological control is another one, and phytoremediation. Okay, maybe introduce water harvesting system in the home. Now with data center using so much of water, maybe encourage home to have water harvesting system where they collect rainwater and reuse back the rainwater to do various activities. Okay, next stop, number 4 is food security. Now, food security, there are 4 aspects. If you can memorize all these 4 aspects, then you can explain all questions related to food security. What kind of question you ask? You ask what is the role of government in ensuring food security? What's the role of student in ensuring food security? What is the impact of climate on food security, like global warming? So these are all chapter 10. They are all possible question 11. So first of all, you need to know what is food security. If not, then how are you going to explain, right? So availability of food means sufficient food for everyone in the country. Yeah? Accessibility means obtain easily. Whether you are living in a kampong or in a bandar, everyone can obtain food easily. Utilization means all the food handler, whether they are the chef or anyone cooking, will know how to make sure the food that they are preparing utilize the best food, the best raw material to cook the best, most nutritious food for everyone. And stability, you can use back the first two words, availability and accessibility. That means food is always available, always accessible. Everyone can get a hold of sufficient food and enough of food for everyone, regardless whether there's a climate change, global warming, whether there's a war. Our country have food for everyone. Okay, everyone will have access to the food, and the food price is stable. You know, like, do we have control price for eggs, for chicken? So food price is stable, food is always available, then that is contributing to food security. Okay. Okay, now 11, 12, 13. Okay, I've got another 10 minutes only. 11, 12, 13 will come out one question, either instruction or essay. 11, 12, 13, I cannot pinpoint what's coming out. If it's 11, they probably ask you to solve a genetic problem. Then you must know the difference between Mendel's law. Okay, Mendel 1, Mendel 2, definition of Mendel 1, Mendel 2. Okay, definition, right? Talking about definition, there are a few definitions you need to remember, not as much as physics, green tech, phytoremediation, eutrophication, Mendel 1, Mendel 2. These are some definition you need to memorize, otherwise you don't get the right term. Okay, like for example, this is Mendel's second law. How do you explain Mendel's second law? Okay, chapter 12. Now, chapter 12, I would say genetic disease. Now, there's plenty of comparison genetic disease. Okay, so now I'm going to do like how to say a tree map for you all to see. Hopefully here got enough space. Okay, so when we talk about variation, right? Variation can be caused by genetic factor and environmental factor. I'm focusing on genetic. So genetic here, you know already, sexual reproduction. I'm using all short form. Sexual reproduction, you have crossing over, you have independent assortment, and you have fertilization. So that is the three event during meiosis 2 and 1 sexual fertilization, which contribute to variation. Okay, if they ask why the cat looks so different from the mother cat, so it's because of this one, okay? Sexual reproduction, crossing over and all that contribute to the different characteristic traits that you see in the offspring. Okay, the other variation is caused by mutation, right? Okay, under mutation, you have gene mutation and chromosomal mutation. Under gene mutation, okay, gene mutation can affect autosome and sex chromosome. Okay, so under autosome, okay, you have sickle cell anemia, cystic fibrosis, and thalassemia. Okay, thalassemia. And then this sickle cell anemia is what type of gene mutation is deletion? Substitution. Substitution, thalassemia is deletion. Okay, and this is insertion, but cystic fibrosis, they normally don't ask. Okay, sex chromosome here got 2, don't forget. Colorblind, and hemophilia. Okay, look here. Sex chromosome here, meaning is linked to X chromosome. There's only 2 diseases. The minute you see these 2 diseases, you must make sure you link it to the X chromosome. Either H to represent hemophilia or B to represent colorblind. So if you see colorblind hemophilia, immediately you link to X chromosome and they fall under sex chromosome mutation. Okay, so in terms of blood disorder, there are 3. 1, 2, 3. Okay, sickle cell anemia is the C-shaped one, right? The blood, okay, looks like a half moon like that. And then thalassemia, the red blood cell is very small because of low hemoglobin. Sickle cell anemia is due to because of the shape is abnormal, right? So the hemoglobin is also abnormal, so it can only transport very low oxygen. Okay, and hemophilia is blood cannot clot. So there are 3 blood disorder. Any of these comparison can come out. For example, this one I put hemophilia and thalassemia. Okay, so they can compare any of these blood disorder. Okay, so that is for gene mutation. Now, for chromosomal mutation, bringing it down here, okay, chromosomal mutation got 2 types, right? One has to do with structural mutation, structure mutation. You have the deletion, duplication, insertion, okay? And then the other one is the increase and the decrease of chromosome, number of chromosomes. So under structure mutation, structural mutated, the structure is mutated, is Cry-Duchet. Cry-Duchet is chromosome number 5, P5. So deleted part of the chromosome number 5. Okay, so this is Cry-Duchet. It is structural chromosomal mutation. Increase and decrease of chromosome can be subdivide into 2, which involve the autosome and also the sex chromosome. So under the autosome, this is the Down syndrome. Under sex chromosome will be the 4 different sex mutation that involve the X and Y. So you have Turner syndrome, Jacobs syndrome, trisomy X, and Klinefelter syndrome. Okay, so I have given you the outline of both the mutation. So with this outline, you will know what is the outline for this. Okay, you will know the difference in term, for example, okay, now you understand what I've just explained. You see, thalassemia occur on autosome. Hemophilia occur on X chromosome. Isn't that what I said just now? This is hemophilia is occur in sex chromosome. This one, thalassemia here, occur in autosome. So with this peta pokok here or peta minda, you can tell already the difference. Okay, so usually, what are the things you need to know is, okay, the aspect is where is it happening? Where? Is it autosome? Is it sex chromosome? Okay, so if the chromosome that is involved, how does it happen? Is it non-disjunction or is it deletion? You know, deletion will be sickle cell anemia. Non-disjunction will be it can be the Down syndrome, the Turner syndrome. Okay, so what type of mutation? The third thing is the number of chromosome. Let's say Klinefelter, 44 plus X, X, Y, right? If it's Down syndrome is 45 plus X, Y, or XX. If it's hemophilia, thalassemia, sickle cells anemia, those they don't affect the number of chromosomes. So it's like us, like normal only. So number of chromosome. Okay, the fourth one is the gender. What type of gender is the person? If it's Klinefelter, is a male. Okay, the fifth one is characteristic. What is the characteristic of the disease? Is it Down syndrome, protruding tongue, slanted eyes? For Klinefelter, it will be small testes, let's say. So you need to know the characteristic, okay? And then for similarity, I think quite easy for you to identify based on what I've explained. So that is chapter 12, okay? And okay, this is the genetic reproduction variation. Okay, I won't go through it. Okay, the last chapter. 5 things you need to know. Okay, don't have time to explain in detail. Okay, what are the 5 things? Number 1 is how do you carry out genetic engineering. Okay, genetic engineering definition also you need to know, right? Example, that how do you produce transgenic potato? How do you produce insulin? How do you produce what does it involve? Okay, so you need to know the steps. Okay, example, insulin. Number 2, DNA profiling. This one is the comparison of the DNA band from the suspect to the victim and find who is the murderer. Number 3, bioremediation. Bioremediation is to use transgenic bacteria to what? Transgenic bacteria will what? They will break down the hydrocarbon in the oil, in the petroleum, into non-toxic substance. Okay, the use of bacteria, genetically modified bacteria. Number 4 is advantages and disadvantages of genetic engineering. Number 5 is, wait, let me see, gene therapy. Okay, gene therapy. From here, these 5 things, I hope I never miss out anything. Let me see. Oh yeah, that is the 5 things. Okay, out of the 5 things, right? DNA profiling quite possible to come out and gene therapy. And then this is the third possibility, advantages and disadvantages. So because these 2 have not come out yet, DNA profiling and gene therapy. Okay, they have not been asked yet, so there's a high chance for it to come up. So, okay, this is okay, this is gene therapy. Remember, gene therapy is the use of a normal gene to replace an abnormal gene using a vector. Now, the vector is usually a virus. So the virus will carry the modified gene or the normal gene into the cell. And then once it's injected into the cell, the new gene will replace the damage or the gene that is mutated inside the cell. Okay, of course, this one you can only do for fetal stage, in fetal stage. And the last one is the process in the DNA profiling. Okay, and make sure you use the word Jalo DNA, or the band of DNA must match the victim one, must match to the DNA band obtained from the crime scene. Okay, if it matches, the band matches, then that suspect person is most likely the murderer. Okay, so this is a very simple process. If you can remember, you'll be good. Okay, so that is DNA profiling. Okay, so why is this such a good method? Because everyone have its own DNA. Everyone have their own set of DNA. So DNA is very unique, and therefore you can be admissible in the court as one of the evidence. Okay, for a murderer lah, the police can submit that in the court as an evidence. Okay, so that's all for Form 5. Okay, so I have another 7 minutes, 8 minutes for you to ask any question. All right, so I'm open to question now. Okay, wait. Carbon sink tu apa? Carbon sink tu, sinki tahu tak? Sinki you basuh baju, you basuh pinggan mangkuk, sinki. So carbon sink is like a sinki yang menyerap carbon. So it's mangrove trees absorb carbon dioxide for photosynthesis. So when you have a lot of trees, in a forest, dia umpama sinki. So we call it carbon sink. Okay? Faham apa lagi? Gerald, good lah. You already answered him. Is it okay if you I personally would not. It depends on the marks lah. Any step for that matter. Any step. Okay, let's say facilitated diffusion got 4 steps. Okay. How teacher know? You go and check it out lah. Got 4 steps. Okay, so step 1, 2, 3, 4. Let's say you write step 1 betul, lepas tu you miss out step 2, tiba-tiba you tak ada step 2, you pergi step 3, step 4 you buat. Okay, so you miss out 1 step, tapi markah 3 markah. So even if you miss out 1 step, you still get your 3 marks, so it doesn't matter. We don't mark in sequence, must have this sequence. We mark it independently. Meaning, if 1 is there, 2 don't have, it doesn't mean you write wrongly already, no. Can we underline the genus if let's say kan, jawapan 1 aja. Okay. Contohnya, namakan organism, okay, rhizobium. Rhizobium is species. Underline lah, underline. 1 aja mah jawapan. Kalau essay, you boleh underline 1. Kalau you rajin, ada banyak masa, you underline lah semua. Yeah, underline. What do you mean by 8 points? Let's say the question is 8 marks. If I give more than 8 marks, will the examiner only read the final? Oh no, of course we will read everything. We read everything that you write. Just make sure you write within the confine of planning lah. Especially question 11, please don't zoom in straight away and write on your essay answer. Plan first roughly in a rough paper. Ask from examiner, petugas bagi you kertas conteng sikit. Okay, kertas conteng. Lepas tu you conteng jawapan dalam kotak. Say, okay, give good, bad to environment, to human. Okay, let's say asap-asap, they draw asap keluar. Okay, kilang-kilang banyak, and then they ask what is the impact or effect of the good and the bad effect on the environment and human if you turn agricultural land into a industrial area. So you buatlah kotak, lepas tu you think apa good effect on the environment. Fikir dulu, tulis dulu dalam kat sini you buat sebagai nota catatan, right? You can just write catat anywhere lah, and then after that you transfer your answer. Is more systematic. Then you will say good to the environment. Okay, garis. Okay, good to the environment. Oh, ni you ada 3 point tadi. Okay, then you tulis lah. Baru cantik presentation you. And then bad to the environment. Bad to the environment. And then, okay. Ada 3 point lagi. Faham tak? Ini untuk you rancang you punya jawapan. Especially question 11. Tolong jangan straight away jawab in your kertas jawapan. Because when the answer is messy, you will lose mark lah. Because the teacher will miss your point. Okay? Oh, extra paper is not provided. Okay, make sure you catat in pencil somewhere and then rub off lah. Think of some planning lah. Catat in pencil, small catatan, and then you rub off before you transfer. Okay. Okay. Mark if we skip 1 step and then go back, can. We mark independently. Is some dependent, but not many dependent. Maybe 1 question is dependent. When I say dependent, it's like this. For example, right? For example, only 1 question usually, A, B. If you say, let's say xylem just now, right? Xylem is hollow tube, continuous hollow tube. And then you after that, ini dia punya fakta. Ini fakta. Explanation dia, you kata dia, to provide mechanical support. Ini salah lah. Macam mana pula hollow tube provide mechanical support? So it cannot, right? It doesn't match. Your fakta does not match with the explanation. Of course, this one salah lah. So your dependent is only one or two questions, but make sure it correlate lah, not like sequential. Sometimes if it's essay, we don't look at the sequence. If you miss one sequence, it's okay. We will still mark the next answer. Okay, any more question? They say we need to make two strategy. What do you mean? They say we need to make a strategy to use the paper. I don't understand that question. Sorry,
Hi,
any
Chikugan.
more? Okay, so if not, then any more quickly? We have one more
Hi,
minute.
hi, Chikugan.
Trial paper are the best for our preparation. I would say it's a good preparation, but it doesn't mean it will come out all entirely from the trial paper. Yeah, the best advice I can give you is this: 60% of the paper will come from your basic knowledge of bio. What is basic knowledge? Chapter 1 to 7 of your Form 4 and chapter 1 to 7 of Form 5. If you can master the basic, you have no problem at all in handling the paper. Okay? So no need to spot so much. The all important is you know your basic. Like if I ask you, what substance or molecule can pass through the phospholipid bilayer? That's called basic. You must know already. What can diffuse across phospholipid? You understand by I mean basic or not? So the first chapter of Form 4 and first chapter of Form 5 is basic and it's definitely coming out. I can spot for you now. It's definitely coming out. Okay, so those are the basic questions that Lembaga want to ensure you must master before you go for your foundation in science or in A levels. Those are the basic thing you need to know. All right. Okay, what are the best trial paper? If you have time, do all. All right. If you have time, do all the trial paper. Okay, any more?
Chikugan,
Try
I think
Melaka.
someone
Okay,
wants.
Melaka is very good paper. Yes, I agree. Melaka is a very good paper. Melaka paper one. Yeah. Paper one now. Okay, by the way, paper one is not going to be easy. Paper one, everyone think, so easy like physics. No, no, no. Bio paper one is tough. Nowadays is tough lah. Okay, so make sure if you can try to do more paper one also. Paper one will help you to remember the facts. It's not as easy, yeah. A lot of students didn't do well in their trial is because of paper one. No, paper two, paper two, teacher already teach so much, they are master of paper two, but then they fail in paper one. Paper one do many questions because it takes 2 to 3 steps before you arrive at the answer. Okay, so that's all. Back to you, Brian. Is that all anyone want?
Chikugan, I
Yeah.
think there's a student who raised his hands.
Oh,
Do you
okay.
have a question?
Yes, I do, Chikugan. For section B, there are two questions to choose from, and we have to only write one. Say I write my answer for both questions. Will the examiner choose the question with the higher mark?
We will mark both. We
Yeah,
will
so
mark
we will
both.
choose the one with the
And
high.
then we will choose the best of the two, the
Okay.
highest mark.
Yeah, very good. Okay, right. Thank you.
Okay. But you make sure you have the time lah. Sometimes you write two, you split your answer until everywhere you did not focus on one, then maybe you won't get as high mark if you were to focus in one.
Yeah,
Okay,
right.
so that's all, Brian.
Thank you.
Any more? Over to you.
Yes.
Okay,
Thank you
thank you
so
for the
much,
opportunity
Chikugan.
to share. Yeah, everyone, I hope all the best to y'all. I hope y'all get A and don't forget to enroll in IMU. Okay, all right, thank you and bye. See ya.
Thank you so much, Chikugan. Okay,
Bye.
so guys, there you have it. Thank you so much, Chikugan. Chikugan have been with us for the past 3 SPM. Imagine that, right? And I'm just
Sorry,
going.
Brian, maybe
Yeah.
they can give some feedback. Yeah, I would also like to know some feedback. Yeah.
Yeah, so guys, maybe you can write down in the chat box on your feedback regarding Chikugan session for today. Also, so I can compile them and share it with Chikugan. Do remember that later at 2 o'clock we have chemistry class by Chigu Lee. Okay, so she is also going to be sharing a lot of important tips and tricks and areas and topics to focus on for your SPM chemistry paper. So I'm gonna end with a IMU video, and I wish everyone all the best for next week's SPM paper. You guys can do well. I believe in you guys, and good luck. Take care.
Before we build the future, we must first care for the well-being of what's already here. For IMU, health is a connected ecosystem. Human, animal, and environment. One planet, one health. As Malaysia's premier private health-focused university, IMU's rich spectrum of programs integrate health with business, technology, and the social sciences, nurturing bold, compassionate leaders committed to building a healthier, more equitable world. Here, ethics is a way of being. We lead with integrity and grow through accountability. We believe that leadership means service, that compassion must come with competence, and every action must lead to meaningful impact. From sustainability to global health equity, we don't just imagine a better world. We work for it together. Because real care goes beyond knowledge. It must also build trust. From pre-university to postgraduate research and studies, our diverse and flexible learning environments foster growth. Across disciplines, borders, and technologies. Because learning doesn't end at graduation. We empower lifelong growth through industry collaboration and translational research that benefit all life in the world. With strong global partnerships, our students don't just learn, they lead, innovate, and serve communities worldwide. At IMU, we believe a healthier world begins with those who care enough to change it.